Okay, so last time talked about anatomy, and the exam's on Wednesday, but it won't cover applied immunology. So I may have to go a little bit faster through this than I want to, but I'm going to talk about some of the basic antibody stuff. This shows you the structure of the T cell receptor and a naive B cell receptor. So you've seen this figure before, nothing new here. Knowing the ultra-specific structure of antibodies, probably not required for the exam, but what you should know is that the constant region is located all throughout here. Anywhere you see a little CH or CL, that's your constant region. And that is what engages the effector functions of the immune system. And then we also have the variable region right here. And the variable region is what binds to antigen. And all of the polypeptides are linked together by disulfide bonds. The antigen binding site is at the very tip. Class switch is going to change the constant region, and the FAB region, which is everything north of the hinge region, is going to contain 3 hypervariable regions, which you can see as these little bars here. And the hypervariable regions are the actual thing that binds directly to an antigen. And so there's a high frequency of mutation in those 3 regions, and they actually come together to bind to the antigen in a 3D kind of way. And the paratope is located, therefore, with those 3 hypervariable regions coming together to bind the antigen. You should know the phrase immune complex, or that term. An immune complex is an antigen-antibody complex. So it's just an antibody bound to a soluble antigen. And immune complexes are generally soluble molecules. There's membrane-bound immunoglobulins, and then there's secreted immunoglobulins. The difference is there's like a hydrophobic segment added on to the membrane immunoglobulins to allow it to serve as the B cell receptor. And when it has that, it's going to complex with Ig alpha and Ig beta on the cell surface. And then when it's secreted, it's called an antibody molecule, and it has a hydrophilic segment, and it will also have a secretion signal added on to the N terminus here at the bottom, and that will be cleaved off when the antibody is secreted from the cell. So the B cell receptor complex has a molecule of immunoglobulin that is membrane bound and monomeric, and it's complexed with Ig alpha and Ig beta, and those contain the signaling domains. They contain ITAMs inside the cell, immunoreceptor tyrosine-based activation motifs. Usually the B cell receptor is going to complex with a couple of other things as well. A trimeric complex made up of CD21, CD19, and CD81. So for CD21, that's just complement receptor 2, and it detects complement fragments. Its ligands are IC3B, C3D, and C3DG. And CD19 is complexed with CD21, and CD19 is an important signaling protein for B cell activation. It doesn't have ITAMs, but it has other motifs that will become phosphorylated on activation. And then CD81 or TAPA1 is also present, and that is a chaperone protein for these other proteins to remain in the correct conformation. Okay. B cells generally require co-stimulation when they take up antigen. They're going to bind to antigen using their B cell receptor, and then you get a signal through the Ig alpha and Ig beta that contain the ITAMs. That turns on a signaling cascade. But you also need CD21, or the complement receptor 2, to bind to complement. Generally considered a required signal, and it sensitizes the B cell to not have to bind to quite so much antigen in order to become activated. So just like T cells when they're naive, just like how T cells have a required co-stimulatory signal, and for T cells B7, B cells also have a required signal, but it's complement through CD21. Okay. We've already kind of talked about this. B cells are going to bind to antigen, take it up, shred it to pieces, load it onto MHC2, and present it to T follicular helper cells. They're going to receive T cell help, and then they're going to secrete antibodies that bind to pathogens and mark them for destruction and engage effector functions. So there's actually 5 specific ways that antibodies provide immune defense. Okay. The first is through neutralization. So antibodies can bind to things like toxins. They can bind to ligands on viruses that are used to bind to cell receptors. They can bind to things like pili on bacteria that bacteria use to adhere to eukaryotic cells and just prevent them from ever being able to interact with our bodies so that they can't do anything past that point. Antibodies can also activate complement. One IgM or 2 IgG molecules will be able to activate the classical pathway. One molecule of IgA can activate the alternative pathway of complement. We'll talk about complement later. They can promote opsonization. So antibody will bind to an antigen, and then the Fc region can be bound by a phagocyte's Fc receptor to trigger uptake and subsequent intracellular killing of the pathogen and the antigen. This can be enhanced by complement fixation in the case of those 3 classes of antibody above, IgM, IgG, and IgA. Fourth, antibodies can induce ADCC, which is antibody-dependent cell-mediated cytotoxicity. So antibodies can deposit on pathogen cells and virally infected host cells, and then Fc receptors expressed on macrophages and natural killer cells and eosinophils can trigger the killing of that target cell that is bound by antibody. So natural killer cells, they're going to recognize IgG fixed on an antigen on a target cell, and they're going to release granzyme and perforin, and that will trigger the death of the target cell. Macrophages and neutrophils and dendritic cells, they're going to release reactive oxygen species and antimicrobial proteins to kill in response to IgG or IgM. And eosinophils can also induce ADCC. That's important for parasite response, right? They're going to release major basic protein. They're going to release other stuff that's toxic to parasites and cause cells to die. And lastly, IgE is capable of arming mast cells and basophils. And then if IgE on those cell surfaces, if it binds to antigen, it triggers them to degranulate and release histamine and other mediators that trigger inflammation and trigger antiparasitic defense. So this is a figure showing sort of a summary of all those things. So there are 5 isotypes of antibody. There's IgG, E, D, M, and A. And a single B cell is only going to make one of these. And in fact, a single B cell will be making antibodies that have a unique amino acid sequence compared to all other B cells. It's going to be of one class and one specificity. So every immunoglobulin made by a B cell is identical, but it will be a unique antigen affinity. No other B cell in your body will have an identical amino acid sequence that makes up its antibodies. One of these isotypes can either be membrane or secreted, and when they're membrane bound, they serve as a B cell receptor. With that being said, naive B cells express IgM and IgD. So all B cells start out expressing IgM and IgD. Okay. And this is a little bit of a more detailed figure showing you the structure of the 5 classes of antibody. These are all soluble secreted forms. IgA, when it is secreted, it is a dimer. So it's 2 antibodies of IgA linked together by a joining chain or a J chain. IgM, it is a pentamer. So it's 5 IgM molecules linked together by disulfide bonds and by a joining chain. Okay. So IgG also has 4 subtypes. You don't need to know the difference between the subtypes or anything like that, or know what they're specialized to do. IgA in primates and humans has 2 subtypes, but not in mice. In mice, they only have 1 type of IgA, and it's closer to IgA1. IgA2 developed in primates first, and then, of course, we are descended from primates, so we have IgA2 as well. So you're going to want to know in detail the 5 classes of antibody and what they do and what they look like, if they're pentamer or monomer or dimer. IgM is the first class of antibody present on naive B cells. So all naive B cells have IgM. So membrane-bound IgM is the B cell receptor for naive B cells. It is a pentamer when secreted, and it's going to be secreted when the cell binds antigen and detects danger signals, which is usually complement, but can also be cytokines from T cell help or from another cell other than a T cell. The secreted pentamer is, as you can probably imagine, pentamer antibodies are really big, so they tie up a bunch of antigens. So they're really good at neutralization. They can tie up 10 toxins at once. IgM is really involved in complement activation through the classical pathway, and that makes it better at opsonizing. And it can be transported onto mucosal surfaces, but in much smaller quantities than IgA. IgG is the most concentrated class in the blood, in the serum. It's made for circulation, but it's also made for tissue immune responses because it can leak out into tissues during inflammation. IgG is the only class that can cross the placenta in people who are pregnant, and it does that through the neonatal Fc receptor, or FcRn. IgG is not quite as good as IgM at complement activation. You need 2 molecules of IgG, but only 1 of IgM. But it can activate complement through the classical pathway. T cell help is required to class switch to IgG, and it can be induced by IL-4, interferon gamma, and IL-21. It is a monomer, so it enters tissues efficiently. It can easily leak out between endothelial cells and diffuse into tissues. And it is a better opsonin on its own than IgM. IgM needs complement activation because IgM is not usually high in affinity. IgG, on the other hand, has usually undergone somatic hypermutation and affinity maturation, so it's really specific for its antigen. IgG is transported onto mucosal surfaces, but again, much less than IgA. IgA is by far the most concentrated class on mucosal surfaces. So IgA, mainly it is a dimer. It can be secreted as a monomer, but mainly it's a dimer. And it's made for transport onto mucosal surfaces. There is a receptor called the polyimmunoglobulin receptor, or the polymeric immunoglobulin receptor. And it is expressed on mucoepithelial cells. It's going to bind to bloodstream IgA. It's going to pull the IgA into the cell, and then it's going to transport it through transcytosis, and it's going to release it on the apical side where the mucosal surface is located. Once it's on the mucosal surface, it binds to and neutralizes antigens, for example, respiratory pathogens, intestinal pathogens, foodborne toxins. That can prevent all of these things from binding to cells to cause disease. So this can coat stuff that could really make you sick when it comes in through your mucosa, like if you inhale allergens, getting an IgA response can prevent allergy. And it can also prevent multiple different intestinal pathogens from causing disease. IgA is also in the blood. It's the second most concentrated class in the blood, behind IgG. It can activate complement by the alternative pathway, and it is induced by IL-5 and transforming growth factor beta. IgE, that is the one that's parasite defense and allergies. Binds to mast cells and basophils through their Fc-epsilon-R1 receptor, or their high affinity IgE receptor, to arm them against parasite antigens. So it allows mast cells and basophils to defend against parasites. It is involved also in allergic reactions. So first it binds to the Fc-epsilon-R1 receptor, then it binds to antigen and gets cross-linked by multiple of the identical epitope on an antigen. And that triggers the receptors to cluster on the mast cell surface. The clustering of the receptors triggers a signaling cascade that causes degranulation of histamine containing granules. IgE is induced by IL-4. And then we have IgD. IgD has somewhat of an unknown function. It's very pronounced on naive B cells. It's expressed as a B cell receptor on naive B cells. And its affinity is going to be identical to the IgM expressed in that same cell. So naive B cells have both IgM and IgD as their B cell receptor. Only very minuscule, tiny amounts of IgD end up secreted, and it's probably by accident. It's probably not intended to be secreted. You're looking at less than 0.001% of circulating antibody is IgD. So it plays basically no role in the immune response other than B cell activation for naive B cells. It's thought that IgD is an especially flexible class of antibody. Its hinge region can bend like all the way back, so it can reach antigen epitopes that are in difficult to reach places and therefore helps with the activation of naive B cells. But of course, that's not something you would be expected to know. Really, IgD doesn't have much of a known function. It's just involved in B cell activation. Just another look at dimeric and pentameric IgM in case you forgot. Okay, so the first way that antibodies can cause defense is through neutralization. Normally, bacteria and viruses can bind to cell receptors, and then they'll get taken into the cell, and then they can infect the cell and cause disease, right? Antibodies can block all the ligands that a bacterium or other pathogen uses to bind to cell receptors and prevent it from getting to those cell receptors. And it can do the same thing to a virus. It can block the viral attachment protein that the virus usually uses to enter the cell, right, and just tie the virus up so it can't get into the cell. That is called neutralization. The virus is no longer able to infect cells. This is another slide on neutralization. In this case, we have a toxin, a typical AB subunit toxin made by a bacterium. The B component binds to receptors, and the A component will be released upon the uptake of the toxin. It'll be released into the cytoplasm where it kills the cell by poisoning it. Antibodies can bind to the B component of toxins and prevent them from being able to bind to cell receptors, so the toxin can never get into a cell. This is another example of neutralization. Okay. And then we have opsonization, which culminates in opsonophagocytosis. Antibodies bind to antigens on bacteria and other pathogens, and macrophages express receptors for antibodies, and that triggers the uptake of the pathogen and the antibodies that it's bound to. And then the macrophage takes it into an endosome and shreds it into tiny little bite-sized pieces, loads it into MHC2, and presents it to T cells. But really, in this case, the immune defense component comes from the uptake of bacteria followed by the shredding of the bacteria. So self-explanatory stuff that I've kind of already been through. One of the mechanisms of defense is ADCC, antibody-dependent cell-mediated cytotoxicity. This is when you have a target that is too large to be ingested, usually. This is also when you have natural killer cells being involved. Natural killer cells are not phagocytic, so they have to kill through some other way. IgG molecules can deposit on the surface of infected target cells. For example, if you have an influenza virus infecting a cell, you're going to express some of the viral proteins on the cell surface, and an antibody molecule can come and bind to those proteins on the cell. And basically, that serves as a signal for a natural killer cell to come and bind to the fixed antibodies using this receptor called Fc gamma R3, or CD16. And that receptor binds to IgG molecules that are fixed on the surface of target cells. It cross-links multiple antibody molecules with multiple receptors. And that is a theme you'll see throughout immunology. You need multiple receptors to be cross-linked and clustering together on the cell surface in order to get an activation response. So you need to cross-link those Fc gamma R3 receptors. And when that happens, it triggers the degranulation of the natural killer cell. And the natural killer cell releases perforin, granzyme, and granulysin, and that kills the target cell. That is ADCC. The same kind of process works in eosinophils, macrophages, neutrophils, and mast cells, also dendritic cells. But in those cases, they're not releasing perforin, granzyme, granulysin. They're releasing reactive oxygen species or antiparasitic compounds in the case of the mast cell and eosinophil. Okay. And then lastly, we have the arming and activation of mast cells and basophils. So IgE antibodies bind to the high-affinity IgE receptor, Fc epsilon R1, on the surface of mast cells and basophils. And then if the antigen comes into contact with the mast cell, the IgE antibodies are going to bind to it. They're going to cross-link receptors and cause the degranulation of the mast cell, usually in the direction of the binding. So usually wherever the antigen is clustering on the cell surface, that's where the granules are going to traffic to before they're released. In this case, you have antigens on all sides of the mast cell, so they're just degranulating 360 degrees, basically, all around the cell. And it also triggers the release of membrane phospholipids through the arachidonic acid pathway. And it triggers the release of cytokines and chemokines that are made from scratch in response to the antigen. This is how IgA is transported onto mucosal surfaces here. There's a receptor called PIGR that binds to dimeric IgA molecules in the circulatory system, and it is expressed in mucoepithelial cells. They bind to IgA, they take up the IgA, they pass it through the cell, and they release it on the side of the cell that is in contact with mucus. IgA sort of binds into the mucus and can bind to bacteria, it can bind to viruses, it can bind to toxins, and prevent them from binding to cells to cause disease. They're just transported out of the gut eventually, and they don't cause a problem. So if you get a persistent IgA response against a mucosal pathogen or a toxin, you can prevent future infection. This is another figure showing you the same thing. I kind of like the depiction of transcytosis here a little bit better, but it's really the same thing. And then this slide shows you how cytokines can induce class switch to different classes. You should know that IL-4 can induce IgE, IL-5 can induce IgA, interferon gamma induces IgG, transforming growth factor beta can induce IgA, and IL-21 can induce IgG. Okay, so those are the 5 things to know on this slide. And then this figure, or this table, basically contrasts and compares all of the different functions and things that different classes of antibody do. So this is a useful slide if you want to have it all in one place. Fc receptors are an important thing to know about. Macrophages have Fc receptors for IgG, IgA, and IgM. They're going to trigger opsonophagocytosis, which is uptake of whatever it's bound to. They also trigger cell stimulation and activation of the respiratory burst. So basically, the cell that bound to the antibody, or binded to the antibody that was fixed on an antigen, it's going to suck in a bunch of oxygen to make a bunch of reactive oxygen species through catalysis. So it's going to turn the oxygen into superoxide anion, etc., to kill the pathogen. And then you should also know the high affinity IgE receptor, Fc epsilon R1, is expressed on mast cells and basophils. And Fc receptors can trigger ADCC. Usual example of this is CD16 or Fc gamma R3, which is expressed by natural killer cells and can trigger them to degranulate and release things that kill target cells. All of this is review, right? And then this chart here from Janeway, I've circled some of the important things to know. Really, it's just a repeat of the last slide. One weird thing is that some of the Fc receptors are actually inhibitory. So, for example, on B cells, it can deliver an inhibitory signal. If a B cell finds that an antigen is already coded in antibody, we really don't need to devote any more resources to producing new antibodies against that antigen. It's already been taken care of. So some of these receptors can inhibit B cell activation. The other receptors are thought to just limit the extent or magnitude of the response to antibody. So they kind of limit how much angry, or they limit how angry immune cells are going to get when they detect antibody. And if you knock out some of these receptors, you get an overwhelming inflammatory response to, sort of innocuous antibodies that aren't bound to anything important. So it's just thought to sort of calm the immune response and, give it a Xanax. I would not say that this content here is important, and it's really confusing for a lot of people. I did include it as an important slide because Sanders in the past asked about it, but I'm going to go out on a limb and say that it's probably not exam 1 content. We're going to look at this in really close detail for exam 2, but this is like lymphocyte genetics and how a B cell receptor is made. I'd wager to say that you don't really need to know this. The basic concept is that when you're making an antibody, you're splicing together for the antigen binding site, you're splicing together a V gene segment with a D gene segment with a J gene segment, and you're just joining 3 random ones together, and you inherit a bunch of them from your parents. And 3 random ones are going to be brought together inside of an individual B cell during development. B cells are going to rearrange their heavy chain first, and heavy chains have V, D, and J segments. So first, D is going to be brought together with a J, right? You're going to pull one of these greens and bring it together with one of these yellows. And then a V segment is going to be brought together with the green-yellow. So you're going to pull one of these reds and bring it together with the green and yellow. And then you have a continuous antibody binding site, basically, and it's going to be encoding a protein. You've made your heavy chain. You've made this blue bar here, this blue chain. Next, the cell has to make the light chain, and there's 2 kinds of light chain. There's kappa and lambda. Kappa genes are attempted first, and kappa genes only have V's and J's. They do not have D's, so this is not present in light chains. So a V and a J are going to be attempted to be brought together, and as long as the receptor works, and as long as the receptor is not self-reactive, the B cell made a successful B cell receptor, and it is allowed to leave the bone marrow where it developed, and it can enter lymph nodes to wait for antigen. If kappa fails, you try kappa on the other chromosome, right? You may have tried it on your mother chromosome first. Now you're going to try it on the chromosome you inherited from your father. If that fails, you can try it at the lambda locus, first on your mother or father, and then you're going to switch to the other one if that fails. So they get 4 attempts to successfully rearrange genes to make a successful light chain. So they're actually really creating this very distal region of their antibody when they do this. The rest of it is already germline encoded. It's just C genes. And C genes don't have anything funky. They're just inherited and used. Okay. We're going to look more in detail at this, so I would not stress yourself out too much about this, but what you really need to know about all this material here is that the heavy chain is rearranged before the light chain during B cell receptor development. The light chain has only a V and a J region, and the heavy chain has a V, J, and D region. Okay? Heavy chain is rearranged first. You bring a V, D, and J together, and then you reach this point. That point, you transcribe it into RNA, and then you splice out all of the introns, and that is encoded into polypeptide. That gives you a heavy chain. Then the light chain is rearranged. You bring a V and a J together. First the kappa is attempted, and then the lambda is attempted, and they're in alphabetical order. And you get to try it on both your mother's chromosome and your father's chromosome. You're going to start in a random place and then switch to the other. So you're going to do kappa from mother, and then kappa from father, for example, and then you'll try lambda of mother and then lambda of father. Obviously, the cell doesn't know which one it's going to try first. It just knows that it's going to go for a kappa first and then a lambda. So you have 4 little regions in your nucleus where there are V's and J's. And you're going to bring together at least one of those. You need to do a single successful rearrangement in order to make a light chain. Once you have rearranged your light chain genes, you transcribe it into RNA, splice out the introns, and that leaves you with mature mRNA that is translated into protein. And now you have a light chain. But again, the only things to really know here is what I have written on the side. Okay. Antibodies, once made during an immune response, really, they're made forever. So if I was to immunize you with an antigen, day 1, I'd see a rapid spike in IgM, and it's going to decline to about 0 eventually. But you're going to see a slower increase in the level of IgG directed against the vaccine. And then if I was to immunize you again, I would see a much higher level of IgG the second time around. The immune system devotes more resources to things that it is exposed to multiple times. However, the IgM response is not going to be stronger. And in fact, it's even going to be weaker the second time around. IgM does not give you memory cells, and it also doesn't give you long-lived plasma cells. So IgG is going to be made for the rest of your life once you're exposed to something. It's probably going to remain at about this level here. But IgM is only made for a short duration after exposure to an antigen or a pathogen. And in addition to that, IgG, it's boostable. IgM, it's not boostable. And IgG lasts forever. IgM only lasts, a few weeks. On the other hand, the antibody affinity of the IgG response increases over time. So B cells mutate their antibody genes, and the ones that have higher affinity are going to be selected for survival. So the affinity increases over time for IgG, but for IgM, the affinity really doesn't increase very much at all. You don't get somatic hypermutation for IgM, okay? It really remains at the same level, but for IgG, you have that whole germinal center reaction happening, so the antibody affinity gets higher and higher, okay? And then this figure shows you the difference between class switch and somatic hypermutation. If your primary IgM antibody looked like this, if it had this blue constant region and this red, variable region, class switch involves changing from this IgM molecule to some other class, maybe IgA or IgG, and now you have a different heavy chain. So the effector function is going to be changed, and the constant region is affected, but only for the heavy chain. Okay, so only heavy chain is altered in class switch. For somatic hypermutation, the heavy and light chains will both be altered independently of one another, so different mutations are going to happen. And the antigen binding affinity is going to change and as a result of this. And only the variable region of both the heavy and light chains are going to be affected. Not the constant region, only the variable region. Okay? So that's the difference between those two things. They both change the antibody. One of them is more random, and that would be this one. On the other hand, this one, you're just switching to a different gene, basically. You've already inherited that gene. You're not changing it in any way. You're just switching to expressing it. And this is sort of a look at the different classes of antibody. I really like this figure because it's kind of aesthetic. But in the human chromosome, I forget what chromosome all of these are located on. You basically have the IgM constant region located first, and then the IgD region. Those are going to be co-expressed in cells that have IgM. And then you can switch to IgG, right? And then you can switch to IgA and then IgE. But you cannot switch backwards because you actually physically delete the DNA that you switch out from. So if originally a cell is expressing IgM and it switches to IgG, you actually cut out all of the constant regions between. So you cannot switch back once you have switched classes. You cannot switch back to a class that you've switched from. In addition, all of the different classes of antibody have different numbers and locations of disulfide bonds, and they have different amounts and locations of carbohydrate attachments on the chains of the antibody. So these little hexagons here are carbohydrates. Okay. The hinge regions are also variable in size. IgA has a tiny hinge region, whereas IgD has a really large hinge region. And the antibodies sometimes have an extra constant region. So you see that IgE has an extra green bar compared to IgG. That makes IgG's constant region a little bit smaller. So they're different in a few ways. And then you've already seen this slide. This is just going to be a reminder slide about how affinity maturation works. But this is evidence of affinity maturation here. This figure shows you the accumulation of mutations inside the heavy chain and light chain in the variable region, which contains the antigen binding site. So you see there's these three complementarity determining regions, or CDRs. These are also called hypervariable regions. These are the regions where the antigen is actually in contact with the antibody. Okay, so these actually physically bind to the antigen. And they undergo the most intense mutation during somatic hypermutation. You can see that during that originally the antibody doesn't have any mutations, but then over the course of 7 days you have antibodies with a few mutations and therefore a little bit of a different antibody affinity. And in fact, it's going to be a higher antibody affinity. If we immunize you again on day 7, you're going to see an accumulation of even more mutations for both chains, right? The light chain is beginning to look really red. All of these little red rectangles are mutations. And the antibody affinity is increasing more and more as time progresses. And then by day 21, when we've immunized for a third time and we look on day 21, we can see that there's literally like 7 mutations here, and each of those can have a really drastic impact on the affinity of the antibody for the antigen. So the intensity of the binding interaction is going to increase. And so basically these plasma cells are dumping out antibodies that are better and better at locating their antigen. And that helps make the immune response more efficient. Really fascinating stuff. And then lastly, we talked about natural antibodies when I brought up the B1 B cells, right? So natural antibodies, they are IgM antibodies made by B1 B cells. They're made from before you're ever born until you die. You don't even have to be exposed to whatever pathogen they're specific for. You constantly make them. Natural antibodies, though, they're usually pretty low affinity, and they're going to bind to multiple things with low affinity. They're going to bind to a bunch of different polysaccharides that are common to different bacteria. They're also going to bind to things like phosphatidylserine that is located on apoptotic cells. That's going to assist in the clearance of apoptotic bodies by macrophages in tissues. That way the tissues stay clear of dead cells and nothing builds up. A lot of natural antibodies are directed against highly conserved pathogen epitopes. Like I said, a lot of different polysaccharides are usually the target of these antibodies. Some of them are specific for self-antigens that you'd normally find inside cells. So if you're finding them in the extracellular environment, that means that the cell has died, and therefore those components need to be cleared out. When a B1 B cell binds to antigen or is stimulated with IL-5, that increases the amount of a natural antibody that is produced by that B cell. IL-5 is made by mast cells, eosinophils, ILC2s, and Th2 cells. And B1 B cells can secrete IgM without T cell help. So they are a rapid response to pathogens that are ubiquitous. However, you can't really get a memory response, and you can't really get the affinity maturation thing with this. So they're limited in what they can do, but they don't need T cells in order to be produced. Okay. So they are thymus-independent antigens. Okay. Now the B cell receptor is going to be taking up these antigens, and co-stimulation can occur in the form of toll-like receptors binding to pathogen components. On the other hand, the normal thymus-dependent antigens, you're looking at B cell receptor taking up an antigen with complement fixed on it, and then it has to present it to a T cell. The T cell has to provide cytokines and CD40 ligand, and then the B cell can finally secrete antibody. The T cell interaction doesn't have to happen in B cells that allow thymus-independent interaction or respond to thymus-independent antigens. They don't need to react with T cells at all. So there's actually 2 kinds of thymus-independent antigens. The only one you should really care about is the Ti2 antigens. And basically these are just repetitive epitopes, repetitive structures like polysaccharides. You can see that here. You've got like a lot of these triangles that are being recognized by these antibodies. And cytokines like BAFF and IL-5 can help promote the production of natural antibodies. So different from thymus-dependent antigens because those require T cell help to produce. So that is B cells. Focus on the slides that have stars. The exam is on Wednesday. I couldn't remember I think I heard something. But yeah, speak up if you have questions. Okay. And then we have to learn about antimicrobial proteins and pattern recognition receptors. Now apparently Sanders was going to do something on toll-like receptors, so I have a little bit of detail on those in here. Again, the starred slides are important. Innate defense proteins. Lysozyme is one innate defense protein. It is an enzyme that breaks down bacterial peptidoglycan in the cell wall of bacteria. It cleaves a glycosidic bond that links together the two components of peptidoglycan, which is N-acetylglucosamine and N-acetylmuramic acid, NAG and NAM. Lysozyme, it's made in the eyes and mouth by phagocytes. It's also made by Paneth cells in the small intestine. And you might remember me talking about Paneth cells during the anatomy stuff. Lysozyme is effective against gram-positive bacteria. It has a harder time killing gram-negatives. And that's because gram-negatives have a cell membrane that surrounds their peptidoglycan. Phospholipase A2, it's involved in a few things, but it can be secreted. It is an enzyme that is basic, meaning alkaline. It can enter bacterial cell walls to cleave lipids in the cell membrane. That can kill the bacterium by opening holes. And it's made by Paneth cells in the small intestine. And that's the main source of secretory phospholipase A2. And then we've got cationic antimicrobial peptides. Now all of these, and the abbreviation is CAMPs, cationic antimicrobial peptides. All of these are secreted as precursor proteins that need to be cleaved in order to become active. And that is to protect host cells from them. So they're synthesized as zymogens. They have to be cleaved. Their pro region has to be cleaved by an enzyme upon secretion into like a gut or onto the skin or into the lungs, right? The cleavage of CAMPs is usually triggered by innate immune cell activation. For example, if a neutrophil takes something up, it's going to fuse its primary and secondary granules, and that will trigger the cleavage of beta defensins into the active form. Or it can be triggered by the secretion of the protein by epithelial cells onto the epithelial surface, like mucosal surfaces or the skin. Besides the lectins, so that's the exception, the CAMPs are amphipathic peptides, meaning they are partially hydrophobic, partially hydrophilic. They have a positively charged or cationic region that is hydrophilic. They have a spacer, and then they have a hydrophobic region that inserts into cell membranes. Okay? The lectins don't have that kind of structure. Instead, they have a carbohydrate binding domain. They are still amphipathic, but they have a tendency to bind to carbohydrates and then insert into the membrane. So they don't really rely on the amphipathic thing quite so much. They recognize carbohydrates. And all of the CAMPs, including the lectins, they infiltrate membranes to open holes that kill the cell, usually the pathogen. Defensins are the first. You can see that they insert the positively charged area is attracted to the phospholipid heads, and then the hydrophobic portion is going to insert into the internal environment of the bilayer. And then if you get enough of them, they can kind of clump together and open up a hole, like a pore in the membrane, and that kills the cell. So these are CAMPs that are 30 to 40 amino acids in length, and they have 3 disulfide bonds. So you're going to want to know the structure of these. Historically, Sanders has asked students about the structure of each of the CAMPs, usually as extra credit, but I can't guarantee it'll be extra credit. Alpha defensins and beta defensins are the two kinds. Alpha defensins made by Paneth cells and neutrophils. Beta defensins made by type 2 pneumocytes in the lungs, and they're secreted. They're also made by barrier epithelial cells in skin and mucosal tissues. And TNF and IL-1 will increase their synthesis. So during the inflammatory response, these are going to become upregulated. And of course, you've seen this slide before, reminding you about Paneth cells. They secrete alpha defensins, and they also secrete lysozyme, they secrete phospholipase A2, and they secrete reg3 lectocidins, which is another class of CAMPs. Then we have the cathelicidins. This is another CAMP, but they have no disulfide bonds, although they do have alpha helices. They are anywhere from 12 to 80 amino acids in length, but on average they are smaller than defensins because they average about 23 to 37 amino acids. They can kill almost anything, including viruses. They are made all the time by neutrophils, macrophages, skin epithelial cells, natural killer cells, and other cells. In neutrophils, they're stored in secondary granules, and they require the cleavage by neutrophil elastase, which is stored in primary granules. So cathelicidins, in order to become activated inside neutrophils, the primary and secondary granules have to fuse together, and that's only going to happen if the neutrophil takes up a pathogen. So it has to become activated for that to occur. Skin and lung epithelial cells secrete these, and in those cases, this cut is going to happen just prior to secretion. Probably all I want to say there. And then we have the histatins, which are histidine rich. That's why they're histatins. They are alpha helical, just like the cathelicidins, right? And they are 24 to 38 amino acids long, secreted into the oral cavity. They are made in salivary glands. They're good against fungi. They have some bacterial activity, and they've been shown to hasten oral wound healing. So this is why things that you wound in your mouth tend to heal a lot faster than wounds that you have on your skin, for example. The lectins are carbohydrate binding proteins made by epithelial cells. One type of lectin is C-type lectins, and they're called C-type because they require calcium to bind to carbs. Okay, C-type lectins are also called lectocidins. They bind bacterial carbs and they form a hexameric pore in the membrane. So 6 of them are required for the pore to form. And one example of lectocidins is the reg3 family. In humans, we have reg3 alpha, and that's reactive against gram-positives. We also have reg3 gamma and other reg3 proteins. And there are also other families of lectocidins. So this table contrasts some of the antimicrobial proteins. Okay, so maybe a good slide to study. And this here lists the locations of each of the antimicrobial proteins. And now we're going to get into the PRRs, the pattern recognition receptors. The first thing to know is this transcription factor. It's called NF-kappa B, nuclear factor kappa light chain enhancer of activated B cells. And it's not a pattern recognition receptor, but it is a transcription factor that's going to be turned on by PRRs. So it's going to be turned on anytime the cell is under intense stress, and it can be made and turned on in almost all cells. It is activated by PRRs, cytokines, or other things like oxidative stress, high pH, low pH, etc. In immune cells, NF-kappa B is important because it turns on a lot of important genes. It turns on inflammatory cytokine genes like IL-1, TNF, and IL-12, and without it, you can't get these produced. It also turns on or expresses enzymes that cause the production of reactive oxygen species in the respiratory burst. So nitric oxide synthase is going to be activated by this transcription factor. It also turns on anti-apoptotic factors to promote cell survival, so that the cell has been stimulated with something that it needs to kill. Hopefully, we don't want the cell to die at the moment, right? We want it to fight off the infection. And adhesion molecules, so that immune cells can interact with each other and so that they can interact with tissue cells, as well as endothelial cells. So NF-kappa B does almost everything we would really want an immune cell to do when it realizes that there is a pathogen. NF-kappa B is located in all immune cells, and it plays a really crucial role in all immune cells. It can turn on IL-2 in T cells. There's just nothing that it can't really do. So macrophages, they're pattern recognition receptors, now that you know what they can turn on. Some of them are phagocytic, others are not. And others are intracellular, which obviously they can't be phagocytic in that case. I'll point out some of the important ones. You've got the FMLF receptor, which recognizes formyl methionine. That is an amino acid that is common to all bacterial polypeptides because it is used as the initiating amino acid during translation in bacteria, right? So that is a structure that is universally conserved across bacteria. Therefore, it's good to have a pattern recognition receptor for it to recognize infection. There's dectin-1, the beta-glucan receptor. It recognizes beta-1,3-glucans. That is common to fungi. A lot of fungi have these in their cell walls, and that triggers uptake of the fungus, or it triggers frustrated phagocytosis if the fungus is too large to ingest. So if the macrophage can't eat it, it's instead going to attack it by releasing reactive oxygen species at it and antimicrobial proteins at it. And it's also going to make cytokines, of course. And then we have Fc receptors and complement receptors. You've heard about those elsewhere. Non-phagocytic pattern recognition receptors include the toll-like receptors. The toll-like receptors are present on a lot of cells, including epithelial cells and innate immune cells. They are transmembrane, as you can see here. Their ligand binding domain is composed of leucine-rich repeats, which are these little trapezoids here. 18 to 25 of those, and the ligand binding domain either faces the outside of the cell or it faces the inside of the endosome, and those toll-like receptors are actually targeted to endosomes. The cytosolic receptor tail down here contains a domain called the toll-IL-1 domain, or TIR, and that enables interaction with signaling proteins. And these toll-like receptors recognize basically anything that you could think of that are present in pathogens. They recognize LPS, they recognize lipoteichoic acids, they recognize flagella, they recognize viral RNA and DNA, etc. They recognize anything you could think of that is common among the different classes of pathogen. Obviously, when a toll-like receptor binds to ligand, it's going to activate NF-kappa B, but it will also turn on the MAP kinase pathway, leading to, increased protein production, upregulation of ribosome components, etc. And it will cause the cell to make cytokines. It'll also upregulate MHC and B7. So this is a pretty picture showing you toll-like receptors. Each of the toll-like receptors has an intracellular domain that interacts with an adapter protein, possibly more than one adapter protein. And then this figure here tells you toll-like receptors. All of them are going to cluster with another receptor when they bind to ligand. Usually they cluster with themselves, so you'll have two molecules of toll-like receptor 3 clustering together on the host cell surface, for example, or the endosome surface. In the case of toll-like receptor 2, it's either going to cluster together with TLR1 or TLR6. And the weird thing about TLR2 is it can also cluster together with dectin-1, which is like the fungus receptor, and it interacts with some of the LPS binding components. The ligand is shown, and the class of pathogen that ligand belongs to is shown, and then you have the cell distribution and the location. What you need to know from this slide: all of the virus detecting toll-like receptors are in the endosome. Okay, so if they recognize viruses, they are in the endosome. If they recognize viruses, they are in the endosome. Everything else is on the cell surface. So if it's recognizing bacteria, it's on the cell surface. If it's recognizing a parasite, it's on the cell surface. Okay. And then you also have the NODs. You have the NODs superfamily of proteins, which is sort of distantly evolutionarily related to the toll-like receptors, except they are soluble. They're not transmembrane. They're soluble in the cytoplasm. Both of them activate NF-kappa B, which is, of course, that classic transcription factor. However, the endosomal toll-like receptors do not really activate NF-kappa B. Instead, they activate neuron-regulating factors like IRF3 and IRF7, and that will promote the expression of type 1 interferon cytokine genes, so the cell will begin making interferons. The cell has detected virus, the cell better make interferons to protect that particular area of the body against potential viral infection. Right? So that is an early response to virus that prevents too much virus from being made. We also have other cytosolic receptors. They also activate interferon regulating factors, and some of them also activate NF-kappa B as well. That's like the retinoic acid inducible gene protein, RIG1, and STING, the stimulator of interferon genes. Those are receptors for viral components, so they turn on IRFs. Okay. Now this slide's not important. This slide's not important. I wouldn't consider this to be muscle material necessarily, but basically the nods are sort of like cytoplasmic versions of toll-like receptors. They're going to bind to usually peptidoglycan components in the cytoplasm, which is indicative of a bacterial infection of the cell, and that will activate NF-kappa B. NOD1 recognizes one component, NOD2 recognizes a different component of peptidoglycan. And then you also have the NLRP superfamily, which forms these really weird structures called inflammasomes. And they recognize a more wide variety of things. So the inflammasome looks something like this. What the inflammasome does, basically, if a NOD-like receptor binds to a pathogen, or a component of a pathogen, it assembles itself into this windmill-like structure here. And that recruits the protein ASC, which recruits caspase-1 to polymerize. And basically, caspase-1 is going to cleave itself into an active form. And then caspase-1 kills the cell. It causes apoptosis. More specifically, it causes a type of apoptosis known as pyroptosis. So caspase-1 carries out the processing of IL-1 and IL-18 into active proteins. The cell is going to die as a result of caspase-1 turning on the apoptotic pathway, but the cell is also going to release IL-1 and IL-18, which will activate the inflammatory response for neighboring cells. So the cell is going to be making a bunch of IL-1 and IL-18, and it's going to release it when it dies through pyroptosis. That's why we call it pyroptosis, or fiery death. It's causing inflammation when the cell dies. Okay. You don't need to know how they form, really, so you can kind of skip over that. And then these are some slides on some receptors that recognize viral components. You've got MDA5, melanoma differentiation associated protein. You've got RIG1. These are sensors for nucleic acids in the cytoplasm. Stuff that shouldn't be there. So like DNA should not be in the cytoplasm. You're probably infected with a DNA virus if you have DNA in the cytoplasm. Or double-stranded RNA, or uncapped mRNA. All of those things are ligands for these proteins. They turn on the IRF transcription factors, and they also turn on NF-kappa B. And then there's this really weird detection enzyme thing called CGAS, or cyclic GMP-AMP synthase. It synthesizes cGAMP, cyclic GMP-AMP, in response to double-stranded DNA in the cytoplasm, a sign of viral infection. And then we have this protein called STING, and it is a sensor for those things that are made by cyclic GMP-AMP synthase. It's going to recognize cJAMP, and then it's going to turn on TBK1. TBK1 turns on IRF3 and NF-kappa B. So same outcome, different proteins. You're turning on IRF in response to a viral infection. Okay? And that is the last of the pattern recognition receptors. And I guess I'll see everyone tomorrow.